Good morning, everybody. Thank you, as always, for joining us. We have a really good talk today. Before that, as usual, we'll go straight into our weekly updates, and we'll start with Dr. Pinsky.
Thanks, Errol, and good morning, everyone. I'll give you an update on the variants in our area. So we have continued to screen all the positives coming through the Stanford Clinical Virology Laboratory. We have seen an uptick in the number of presumptive B117 or UK variants. In addition, the California variant, or 427-429, with the L452R mutant, remains highly prevalent, about 60% of our cases. While the case numbers have been relatively stable and low in our area and other parts of the world, the number of cases are increasing, particularly in India, where they're having a spring surge. There is a new variant that's been reported out of India. This contains the L452R mutation, which is found in the California variant, as well as E484Q. This is called a double mutant by the BBC, and that has sort of caught on. If you'll remember, the E484 position is mutated in a number of these variants of concern and variants of interest, except to a K. In this case, it's to a Q, or glutamine. You can see here the increase in number of cases. In India, this is a 55% increase over the last week, and much of this is occurring in the Maharashtra area, state of India, which includes Mumbai. So we noticed about a week or two ago these unusual curves in our mutation-specific real-time PCR assay that looks for these common mutations. This is the probe for E484K, and you can see that nice sigmoidal curve crossing here the threshold and our famous CT values that everyone is now incredibly familiar with. But you can also see this E484Q mutant that is sort of flattened. So we saw these and then sequenced them and determined that they were this new India variant containing this particular E484Q mutation. And this probe has one base pair difference between K and Q. So as I said, we sequenced this mutation, this strain. It is a G-clade B lineage virus. There are not just 2, but 8 non-synonymous mutations in the spike protein, but these are the important L452R and E484Q in the receptor binding domain. Here is a phylogenetic tree showing our sample here. And all of these ones in the light blue dots are primarily samples that were submitted last week to GIS AID. From India. So we're clustering with all of those new cases. Importantly, this strain did not arise from the California variant, even though it contains the same L452 to R mutation. So we are now detecting this Indian variant in our patient population. We have confirmed 1. There are 7 presumptive that have this unusual PCR curve in the presence of L452R. So this demonstrates the rapid spread of these variants throughout the world. And I believe that these are the first of this Indian variant reported in the United States. So we will keep monitoring this and keep you updated on a weekly basis. Thank you.
Dr. Pinsky, as well, thank you very much for those updates, and thank you for keeping us updated in future grand rounds as well. Moving on now, I want to first just briefly mention next week we will have, this is a part 2 of kind of a part 1 that we'll have this week talking about long COVID. Drs. Subramanian and Williams will be joining us, focusing more on the clinical aspects and the work they're doing on long COVID. The week after that, we'll have Dr. Michael Liu. He's the Dean of Public Health in Berkeley School of Medicine. Some of you may have seen this very impactful email that he sent out last week. And it was called essentially addressing the violence against Asian Americans and Pacific Islanders. And we're really excited. We quickly invited him and he quickly responded. And so we're really excited to have him present the week after that. And the week following that, our own Dr. Parsnett will talk to us more about herd immunity. We're all excited to get to. But for today, we have Dr. PJ Utz. Dr. Utz is a professor of medicine here at Stanford University. He's the Associate Dean for Medical Student Research and Scholarship. He's a Director Emeritus of the Medical Science Training Program, MSTP program here at Stanford. He's a Program Director for the Accelerating Medicine Program Partnership Leadership Center for Rheumatoid Arthritis and Systemic Lupus. He's the Program Director for the Stanford Explore Summer Students Program here at Stanford, and is the founder and director of the Stanford Institute of Medicine Research, or SIMR. It's a summer student program here. It's one of the nation's largest high school research programs devoted to improving diversity in our nation's biosciences and physician sciences workshop. As you can see, Dr. Utz has a huge commitment to educating and being involved with the future generation of our clinicians and researchers. He has a very active research lab studying autoimmunity, infection, vaccines, and technology development. Just looking at Dr. Utz's CV, he's got 9 active grants from the NIH, Bill Melinda Gates Foundation, and many, many more grants pending. He's extremely active. We've known this for years from our communications team, always covering all the stuff that he does. Recently in January, Dr. Utz was the senior author of a preprint article that came out called The New Onset IgG Autoantibodies in Hospitalized Patients with COVID-19. And this was an article that was done in conjunction with a number of other authors, particularly many faculty from our own here at Stanford. And it got a lot of attention and continues to do so. And from the communications group, we see all that. And Dr. Utz is extremely busy. People want to know what's the pathophysiology of long COVID, and he's been in the news a lot with this. We asked him to speak at Grand Rounds today to help us all understand better the aspects of long COVID. So Dr. Utz, I'm going to stop sharing my slide here so you can start sharing yours. I just want to say thank you for being with us today, and thank you for all you're doing to help us, for all you've done and research with autoimmunity to begin with, and particularly now with long COVID. Thanks so much for being here today. One last thing before I send out Dr. Utz, I just want to, just a quick update to everybody. We have a survey that's going to go out right now, a couple questions for Grand Rounds. If you don't mind taking a look at that, and we'll send out the email as well. So please take a look at that survey. It's a couple questions to help us improve Grand Rounds for you. Coming back to Dr. Utz, thank you so much for being with us, and I'll turn it over to you.
Great, thanks. Are you able to hear me okay?
Yeah, perfect.
Excellent. So I give enough of these talks to know that I frequently have problems with my audio. And so if I lose it during the talk, signal me in some way. So thanks again for the opportunity to present today. And I'll start off just by disclosing conflicts of interest. I'm on SABs and have sponsored research and own private stocks in some companies that are related to COVID, developing COVID therapeutics or diagnostics. None of that is really relevant for today's talk. So the story here is that my lab has been working on autoimmune diseases for decades and technology development. And when in March rolled around and labs got shut down, we had 3 months to really think more about this disorder. And I was starting to hear reports from clinicians taking care of patients here at Stanford and elsewhere describing manifestations that to me sounded like autoimmunity. Some people were describing patients who had what sounded like new onset arthritis, not just arthralgias, but arthritis and vasculitis and other manifestations that suggested maybe there's an autoimmune component to this disorder. The list of potential autoimmune and inflammatory manifestations in COVID goes way past this slide, but some of the most common ones that common meaning that you've probably read about these or certainly seen papers on them. One is MIS-C, this disease that resembles Kawasaki's disease in children that's also now been described in adults. A very significant proportion of patients develop arthralgias, at least a third, and about 50% develop myalgias. But some patients have gone on to develop full-blown myositis that's associated with autoantibodies, and some also have myocarditis. There are a number of different CNS manifestations, and you'll hear more about those in next week's Grand Rounds. And another common complaint is sicca symptoms, which is something that it's observed in patients who have Sjogren's disease. Vasculitis has also been pretty commonly reported, and we'll go over some of the autoantibody tests that are associated with ANCA-associated vasculitis, or anti-nuclear or anti-neutrophil cytoplasmic antibodies, or so-called ANCA. And then finally, thrombosis. And this has also been a pretty hot topic, as I know that those of you caring for COVID patients in the hospital are seeing patients who have thrombosis. And there's a very elegant paper that I'm going to go over that talks more about these autoantibodies. So with this as a basis, my lab started to work on this late in May, early June, and then a series of papers started coming out describing autoantibodies. And so let me just take you through a little bit the history of what's already out there, some of the seminal papers, and what I'll then describe as an update where we've now gone after the actual antigens, the targets of the immune response in patients who have COVID-19 who develop autoantibodies. And some of what I'm going to show you, I think, is very consistent with the literature, and some of what I'll show you, I think, refute some of the things that are in the literature. So the first paper that came out was from a group in Greece, published in the Annals of Rheumatic Disease in, I think it was late May. And they looked at 29 critically ill COVID patients, all of whom had been hospitalized. A number of them ended up dying. And what they found was that there were a pretty significant proportion who had anti-nuclear antibodies. So when somebody comes in with a potential autoimmune disease, one of the tests that's done is an ANA to just see if they could have anti-nuclear antibodies. And what they found was that about a third of the patients had them, and many of these were in a nucleolar pattern. So remember that when you order an anti-nuclear antibody test, the traditional way, using fluorescence, cells are on a microscope slide. You add serum from the patient, and if there's antibodies that bind to the nucleus, those are detected using a secondary antibody that is conjugated to a fluorophore, so the nucleus glows, and it glows in certain patterns. And so one of those patterns is a nucleolar pattern. I'll actually show you an example of that from our own study in a moment. But this is atypical. Nucleolar is not the sort of pattern that you see commonly in patients who are ANA positive. And so this was a tip-off right away early on that something is different about patients with COVID who develop these autoantibodies. They reported also that 14% developed ANCA-associated antibodies. And in some of these cases, the antibodies were associated with clinical findings suggestive of vasculitis, not in all patients, but in some of them. And finally, they found that about 40% of patients had antiphospholipid antibodies, either IgG or IgM. This is also a very large proportion of those patients. Since then, several other papers have been published, one in Cell by Gruber from Mount Sinai, looking at antibodies in MIS-C, where they did a screen using a phage library approach. And they found antibodies against the LA protein, which is also called SSB. This is targeted in patients who have lupus, Sjogren's, neonatal lupus. And then Inaki San's group at Emory published a paper in medRxives in the fall showing that 44% of their patients had positive anti-nuclear antibody tests. And many of these ANA titers, a significant proportion of those patients had very high titers of ANA. Now here at Stanford, I remember relatively early in the pandemic talking to Scott Boyd about this, and Scott said that he had run some ANA tests on Stanford patients and was not seeing a lot of reactivity, or if anything, there was weak reactivity. And as you'll see in a minute, that's one of the first tests that we ended up doing. And we do not see in our study ANA titers that are high titer, nor do we see numbers approaching 30 to 40%. So a really important paper came out in the fall from Jason Knight's group in Michigan. And they showed that in 172 patients, these are all hospitalized patients with COVID, 52% had antiphospholipid antibodies. And they added all of these up using, this is a commercial kit, that's a commercial kit. And they looked at IgA, IgM, and IgG antiphospholipid antibodies and added it all up. And they found that half of patients had these antibodies. They went on really elegantly to show that these antibodies are directly pathogenic, both in vitro and in mouse models. In vitro, they cause neutrophil extracellular traps or nets. I'll show you an example of that in a moment. And they actually cause clots in an animal model. So these are clear. Whether there's really 50% or not is an open question. I think that number is strikingly high, and I don't think that the numbers in reality are that high. but I think their data pretty convincingly show that these antibodies are pathogenic. So one of the first things that this group did was that they took neutrophils from healthy individuals, and you can see those here, they're labeled with DAPI, so these are just the nuclei of those cells. And they added control immunoglobulin from healthy patients. And as you can see, nothing really happens to these cells. If they do the same thing with a patient with severe COVID, so they purify the immunoglobulin from these patients and they throw it on these neutrophils, now you see the neutrophils explode. There's different types of cell death. So there's necrosis, there's apoptosis, and this is a special form of cell death called netosis. Netosis has been around for a long time. In fact, one of the earliest descriptions was in patients with malaria, where you could see netosis occurring in blood smears from patients. And so what they found was that if you add this immunoglobulin, then the neutrophils explode and they extrude their chromatin. You can see these long lines of DNA and chromatin, and embedded in the chromatin are inflammatory peptides that are actually defensin type peptides that are thought to help eliminate microbes. And this chromatin also wraps around organisms and kind of keeps them in place. These are highly inflammatory. They cause the release of lots of inflammatory cytokines. And so this is the first step in what they think is a process that then goes on to develop clots. And so they showed this in a mouse model where they took mice and they inserted a catheter into the inferior vena cava. They used electrolysis to damage the endothelium. And then immediately after that, they infused purified IgG, either from healthy control subjects, shown here, patients who had severe antiphospholipid antibody syndrome. So these are patients who have what's called catastrophic APS syndrome. And for those of you who have seen it, it is really awful. These are people who have widespread clotting. They necrose off their fingers and toes and limbs. It's just awful. They also looked at patients who had high levels of antiphospholipid antibodies who had COVID and those who had low levels. And 24 hours later, they just went in and they looked at the inferior vena cava. And you can see that the control subjects really didn't have very much in the way of clot. Patients with catastrophic APS had very large clots, as did patients who had high levels of antiphospholipid antibodies. And then patients who had low levels of autoantibodies had relatively smaller clots. They then eluted out the antibodies from these clots, and they showed that these antibodies bound a complex of myeloperoxidase, which is found in the neutrophil granules, and DNA. So this is a readout for the presence of nets that would be found in the clots. And they showed that, again, in the catastrophic and in the high patients with high levels of antiphospholipid antibodies, when you look what's in their clots, sure enough, they have these nets. So when you take all of this together, this provides very compelling evidence that in a subset of patients who have severe COVID-19, typically hospitalized, and especially those who develop clots, this is almost certainly the mechanism or one of the important mechanisms by which these patients are developing an immune response against a self molecule that then leads to a clinical phenotype. So we decided to test several different hypotheses in our studies. And so first is that specific autoantigens are targeted in COVID-19. It's been well known for a while that when you have a viral infection, whether it's influenza or severe rhinovirus, you can develop low-level ANAs that tend to be transient, and nobody really knows what those target molecules are. And so we hypothesized that there are specific antigens that are targeted. We also hypothesized that in some patients, they already have these autoantibodies at baseline. And that raises the question, if we find autoantibodies in patients who had COVID-19, and I'm going to show you that that's what we have found, there are autoantibodies that are found much more prevalently in COVID-19 subjects than healthies. Does this mean that they have autoimmunity as a risk factor for developing severe COVID? Or alternatively, do they develop autoantibodies that are brand new? Some of which, like I've just shown you, could be pathogenic, and we hypothesize that these are directly triggered by the virus that causes COVID, SARS-CoV-2. So this paper was really a tour de force. We started doing work in May or June. And part of it was to try to contribute to the field. We were getting sick of sitting at home waiting to get back to the lab. And part of it was just, was out of necessity. We wanted to get the lab up and running again, and there were grant applications that were available. And so we quickly pulled together a group, and this is all of the people from Stanford who are co-authors on this paper. We actually got the reviews back last night. It got very good reviews from Nature Communications, so we're pretty confident that it will be published. We just have to revise some of the wording, no new experiments. And I'll point out that this is a large number of people who contributed to this study in many different ways. And often at Grand Rounds, you'll see people like myself or Catherine Blish or Taya Wong or other people presenting their data, but behind the scenes, there are tons of people who are caring for patients, collecting clinical data that allow us to go back and study these samples and to do clinical correlates. Without all of these people, none of this would happen. At the end of my talk, I'm also going to point out that this is, it's a special day, I guess, in many ways. It's Cesar Chavez Day, and it's also the last day of Women's Empowerment Month. And so as I go through this talk, I'll point out that a majority of the names that you see here are actually women. And these are the main people who contributed to the data I'm going to show you now. So Sarah Chang is a star research assistant in my lab who's going to be unfortunately starting at Genentech on Monday. Alan is an undergrad who was banished to Alabama for a year and just got back. He's now back in the lab and was doing data analysis at a distance. Wen Zhao and Socrates at Penn worked directly with Nina Luning-Prock, who's one of the corresponding authors together with Chrysanthi Skavaki in Marburg. And Chrysanthi and Nina were instrumental in pulling all of this together. Sharia newly joined the lab and performed the blocking assays that I'll show you in a minute. So again, the theme here is there are actually more women than men who contributed to even the corresponding author and the first authors. So the first thing that we did was we got as many COVID samples as we could get. And Stanford, one of the reasons I love being here, many of you know I trained at the Brigham, great place to train, reasonably collaborative, but not like it is here. Stanford is one of the most collaborative places that I've ever been. And so once we decided we were going to try to do this study, people jumped out of the woodwork and wanted to collaborate. And so Taya was the first. She provided 48 samples from patients from Kaiser Permanente. We had no clinical data on these patients there and no longitudinal data. We got a number of samples from Penn and Marburg. These are particularly important samples for us because together with the samples from Kari Nadeau and the LTI clinic, because these are longitudinal samples. We had samples at more than one time point, which allows us to test the hypothesis that some of these autoantibodies are brand new, and also to determine what patients might have pre-existing autoantibodies and what they might be. We've been working with UPI and PROS on their interferon lambda trial. I know PROS presented this in the past. And this is also a phenomenally helpful study because this was done in subjects who had mild COVID, were very early in their disease process, and they've been followed now for probably 9 months or longer. And so this also allowed us to look at several different time points. I'm just going to show you one data slide from this study. We also got convalescent plasma samples from Nina. So these are patients who had mild COVID, came in 28 days later and provided plasma for therapeutics for patients. And then we had a bunch of healthy controls. So altogether, we looked at almost 200 samples in this study. So the first thing that we did was we looked at the ANAs. And so a problem with ANAs in general is that depending on who's doing them, what lab is doing them, if they're not expert at doing them, you can get all kinds of different results. So Nina is an expert. This was done at Penn. She runs the clinical lab there. This is a CLIA certified assay. This is exactly the assay that you would order if you saw a patient in a rheumatology clinic. So she ran these, Wen Zhao actually ran them. And what we found was that about a quarter of all of the patients had anti-nuclear antibodies, and they had all kinds of different staining patterns. So some of them were homogeneous, like this patient Some were nuclear, like this patient, nucleolar. So this is somebody, this type of staining pattern is what you might see in patients who have systemic sclerosis. And then this is a typical speckled pattern that you'd see in patients who might have lupus. We also saw patients who had cytoplasmic. So some of these patients negative with cytoplasmic. That's suggestive of antibodies that one would see in patients with myositis. And we'll see that in a minute. Patients with myositis often are ANA positive in a cytoplasmic pattern. So technically they're ANA negative, but they have antibodies against proteins in the cytoplasm. And that's a good thing for house staff to know about and to keep in mind when they see patients with a cytoplasmic pattern. You should think about involvement of muscle in terms of the autoimmune manifestations. So about 25% of them are positive, and they're pretty weakly positive. They were all in the order of 1 to 80 to 1 to 160. Marco Radic and Wen Zhao, so Marco is at the University of Tennessee, also collaborated with us on this project. And at Penn, they did ELISAs for RBD and nucleocapsid. So you can see most of these subjects have pretty high levels of these antibodies. They looked at double-stranded DNA antibodies, antibodies against myeloperoxidase, and antibodies against proteinase 3. These two are associated with ANCA-associated vasculitis. And unlike what the literature shows, virtually none of these patients had positive MPO, or in fact, actually none of them had a positive MPO or PR3 with the exception of this one subject. And this subject did not have clinical vasculitis. So the take-home point from this slide is that the common antigens that we might look at for vasculitis or lupus do not really seem to be targeted. This is a photograph of a slide from Judith James. So Judith and myself and about 150 different people participated in a 2-day meeting hosted by Tony Fauci in December on post-acute sequelae of COVID. And it was an eye-opening meeting for me, for sure, because I had not appreciated the breadth of the different disorders that patients who have COVID are, what it's like for them after they recover. And so one of the things that Judith did, she's at Oklahoma Medical Research Foundation. She also runs their clinical immunology lab and is an autoantibody expert, one of the world's autoantibody experts. She looked at 113 COVID patients and looked for specific antibodies in CLIA-certified assays. And she found similar results. So there's actually very few patients who have antibodies against typical things like double-stranded DNA, RNP, centromere, and so forth. And importantly, she also found only 4 subjects out of 113 had antibodies that would be associated with antiphospholipid antibody syndrome. And so although the Michigan study showed 50%, I'll show you in our study in a non-CLIA certified, just a research grade assay, we found probably between 15 and 30%. I think that's an overestimate, and they did not correlate with clinical findings. She also finds a very small number of patients with these antibodies. So the conclusion from the first part of this talk is that if you're screening patients using really good CLIA level assays, only about a quarter of patients are going to have an ANA. This is probably high. It's definitely higher than you would expect in a general population, even in an elderly population, which is the hospitalized population in general. They tend to be low titer. They have all kinds of different patterns, including patterns that we don't see really commonly. And then the common antigens that you would test are usually not prominently identified. So what we decided to do was to develop a screen using a Luminex bead-based assay. And this is something our lab has been doing for 2 or 3 years. Bill Robinson started protein array development here at Stanford almost 20 years ago when he was a postdoc in Larry Steinman's lab and he was working with my lab. And so what we did was we created an array of 73 traditional autoantigens. Many of these are things that you wouldn't test in a regular lab, so they're special antigens. We also looked at cytokines, chemokines, growth factors, specifically to look at secreted proteins, because as you'll see, one of the most important groups of proteins that are targeted in patients with COVID-19 are actually secreted proteins that you'd never pick up in an ANA test or a traditional assay. And we also developed a viral array that I'm not going to talk about. This could not have happened without help, in particular from Peter Kim and Taya Wong's lab. They provided us with many of the antigens that were being made early on, and then provided to Scott's lab, who's gone on to develop more of these antigens. So the principle of this is pretty straightforward. We take these microbeads and we couple to the surface of the microbeads different antigens. So each bead has a different antigen on it. We then mix, in this case, serum or plasma. And if there's antibodies in there, those antibodies bind to the antigens that are found on the surface of the bead, shown here. And then we detect this bound antibody using another antibody that glows. So it has a phycoerythrin on its surface and it glows. And then this goes into an instrument that then reads what the bead is, which tells you what the antigen is that's on it, and also measures the fluorescence intensity, tells you how much glowing there actually is on the bead. So we create these custom arrays just by putting different molecules into a microtiter plate, conjugating the individual beads. We can do 96 of these at a time, but we can actually measure 384 different proteins at any point in time. And then it goes through this instrument, which we got certified for doing COVID studies in the lab. We went through a lot of discussions about what antigens to include on the array. And we tried to focus on things that we thought would be relevant to COVID. So things associated with arthritis, cytokines, obviously, some thrombosis-related antigens, complement proteins, and then many antigens from lupus and scleroderma and myositis, because these antigens have RNA molecules in them. And so the thought process there is that, that this virus could trigger an immune response specifically against proteins that are associated with RNA molecules. And I won't go into the molecular mechanisms for why we think that's the case. So this is the take-home first figure from the paper. And so I realize that it's small. These are the antigens here. These are the patients here. And all you really need to know is you're looking for red. These are COVID patients here. These are healthy controls here. And we measured the mean fluorescence intensity. We calculated what the mean was for healthy controls. And then we took 5 standard deviations above that. So we went way above the traditional cutoff, and we were just looking for screaming hot results. And what you see here is a lot of red. In fact, about 50% of patients with severe COVID had 1 or more autoantibodies when compared with healthy controls, where we found, in this case, only 4. And the typical antigens that we find in healthy controls are thyroperoxidase or thyroglobulin. That's because autoimmune thyroid disease is just very prevalent in the general population. You'll see that these antigens, we'll go over these in more detail in a moment, but they cross a lot of different areas, but especially scleroderma and myositis antigens. So let's first look at antibodies that are potentially pathogenic or protective, as the case may be, we don't know. So first are beta-2 glycoprotein 1 antibodies. These are healthy controls here, prototype immune samples from patients with lupus or related diseases here, and these are all the COVID samples. And you can see that there's generally higher levels of these antibodies. A number of them cross the 3 standard deviation threshold, and about 6 or 7 cross the 5 standard deviation threshold. We found 2 subjects with very high MFI. So this particular subject is at 20,000 MFI, that's very high, who had antibodies against the complement protein C1q, which I'll show you in a minute. This is an important protein for getting rid of immune complexes. And so antibodies against C1q have been described in lupus and correlate very strongly with renal disease and in particular, severity of renal disease. And we also identified 2 subjects who had these super interesting antibodies against ACE2. So we don't yet know whether these antibodies are blocking antibodies or if they somehow potentiate the binding of a virus to ACE2, but these were also in very high levels, 15 to 20,000 in this subject, and also in the second one. None of the other patients or healthies had them. IgM anti-ACE2 has been described by Anthony Rosen's group at Johns Hopkins, and anti-ACE antibodies were described years ago in scleroderma. So this is an interesting finding. We don't yet know the significance of it. So just to show you one example of something that could potentially be pathogenic or alternatively could be protective, depending on how you look at the biology. Anti-C1q antibodies bind to the tail, collagen-like tail of C1q, which binds to these immune complexes and is responsible for clearing immune complexes. And so this is a slide provided by Bill Robinson. He's been collaborating with Cynthia Magro and Jerry Nuevo, and they have very, very elegantly shown that if you biopsy skin from COVID patients, a severely ill COVID patient, even skin that looks pretty normal, and stain, what you find is you can find ACE2 upregulated in the endothelium of the skin. ACE2 is itself an interferon-inducible gene. And so one hypothesis could be that with all of the interferon floating around, ACE2 is upregulated in the endothelium. They're able to find virus that's also present, but the virus lacks the RNA. So they're pseudovirions. They can find the spike protein and nucleocapsid, but not the RNA. There's obviously antibodies there, and they also find C1. And so the anti-C1 that we see could work in a pathogenic way by preventing the removal of these immune complexes, or alternatively, it could be protective in some way if it's preventing the activation of C3, C5, and then endothelial and microvascular damage. So some of the main target antigens that we see are molecules that are found in, that are targeted in myositis. So myositis is inflammation in the muscle, relatively rare disorder. And as I've already said, these patients tend to have anti-cytoplasmic ANA staining patterns. They'll make antibodies against the signal recognition particle and especially against the tRNA synthetases. So remember from your basic biology that DNA is made into RNA, which is made into proteins. And the way that the protein is made is the ribosome comes in and reads the messenger RNA and uses tRNAs to bring in the charged amino acids to construct the polypeptide chain. And so there are roughly about 20 tRNA synthetases. These are targeted in patients who have myositis. So what we found is this is an example of 4 different tRNA synthetases that we found that were targeted across these subjects. And so one is EJ, PL12, JO1, which is associated also with the Rho protein in mechanics hands, and then PL7. This subject actually had an increase from roughly about 15,000 to 20,000 between day 0 of hospitalization and day 7. There are other myositis antigens that are targeted, but I don't have time to go over each of them. We also found Scleroderma autoantibodies. And so we see antibodies against centromere protein A, centromere protein B, and we had 2 subjects that had pretty high levels of SCL70 compared with a prototype Scleroderma patient. And interestingly, the anti-centromere antibodies in 2 Stanford subjects had no anti-centromere antibodies in the first sample, and then very high levels 7 days later. Same thing here, nothing here, and 7 days later, very high levels. And this again demonstrates that at least in a subset of patients, they develop antibodies that are new that were really not present prior to the infection with COVID-19, as best we can tell. So what happens over time? And I'll just tell you that most of these antibodies are actually present at baseline, but as I just showed you, many of them are not. So here's a few examples of some that are present at baseline. Here's an SCL70. All of the anti-thyroid antibodies that we have observed, which is roughly about 12 to 15% of COVID patients, all of them that we have, where we have paired samples, were present at baseline and really did not change at all. And even the anti-C1q antibodies seem to be present in the first available time point. So it's possible that these may be maybe they're already and perhaps are serving in a pathogenic role for just selecting out those patients who have the bad antibodies and they're going to have a worse COVID course. We'll have to literally study thousands of subjects to really determine if that's the case. And these are just some examples of antibodies that I've already shown you some that go up, but we also have some that go down. This MDA5 subject was present at the first time point, went away. PL7, we had 1 subject who had 3 time points. They were elevated, went up, and went down. So this gives you the suggestion that some of these autoantibodies might actually be transient. And I think to really determine all of this, we're going to need to study subjects over a period of probably at least a year to determine whether what was there before or early on in the infection, what was there when they were really sick, and what was there later on, perhaps explaining post-acute sequelae of COVID in some patients. So the second conclusion here is that we are not finding common antigens, but rather rare antigens targeted in scleroderma, myositis, and some other disorders. We think some of these are likely to be pathogenic. Many of the antigens are RNA binding proteins. Some are fixed, others are transient, and some appear to be newly induced. So let me just go to the last part of the talk, which is the anti-cytokine antibody story, which is also super fascinating. So the take-home point, if you take nothing home from this talk, is that some infectious diseases are fundamentally not infectious diseases at all. They are autoimmune diseases. And I've shown this before at a grand rounds, maybe 5 or 6 years ago, but this is a beautiful study done by Steve Holland, in which they had a group of 35 patients from Southeast Asia who had disseminated mycobacterial infections that they could not clear. And they did not have genetic defects. And Steve went on to show that they had anti-interferon gamma antibodies. And moreover, if you gave these patients steroids and rituximab, which you can imagine what a malpractice attorney would think of that in somebody with a disseminated mycobacterial infection, you can actually clear the infection. So it's very clearly an autoimmune disease that leads to an infectious disease. And this is from their New England Journal article that shows other infections they can get. So these are the atypical mycobacteria. Here's a patient with cryptococcus. Here's a neutrophilic dermatosis. And it turns out that these anti-cytokine antibodies are much more prevalent than you might imagine. And they do all kinds of different things. They have been associated with pure red cell aplasia in patients who have anti-EPO antibodies. Patients with pulmonary alveolar proteinosis can develop antibodies against GM-CSF that are thought to be pathogenic. There's antibodies against interferon alpha, IL-22, IL-17, and lots of others that I'll show you in a minute. We have also previously shown that these occur in patients with scleroderma and lupus. So we've identified anti-interferon antibodies in patients with lupus, as well as anti-BAFF, which is a B-cell activating factor. So one of the most important papers I think that's come out of this whole COVID pandemic is this really, really cool paper by Jean-Laurent Casanova. And they screened over 1,000 subjects, or around 1,000 subjects. And they identified that patients with life-threatening COVID-19 have antibodies against interferon alpha. About 12% of men, only 2 to 3% of women. And if you have mild COVID, almost, well, none of their patients. We have some patients with mild disease who also have them, have these antibodies. And so their hypothesis is that in a population of millions of people, some people have anti-interferon alpha antibodies and they're walking around with them. If they get infected with the virus and you don't have these antibodies, you tend to do fine. If you have the antibodies bind to interferon alpha and they prevent binding of interferon alpha to its receptor called IFNAR. And that blocks the signaling cascade that activates this cell and tells the cell, hey, I've got to now get rid of this virus. So it doesn't do so, and the patients have very poor outcomes. So this is an example of an autoantibody that would appear to be potentially pathogenic and present prior to the development of severe COVID. In fact, they say that they think that this is what's causing severe COVID in that 10 to 12% of men, and that these autoantibodies are always pre-existing. I'm going to show you that for interferon alpha, that's probably the case, but it's definitely not the case for other anti-cytokine antibodies. So we again screened the same patients as the same side of the figure that I showed you earlier. This side is the anti-cytokine antibodies, and what you see here is a ton of red. So these are interferons here, interleukins here, and other proteins here, including another ACE2 positive subject here. So the take-home point here is you don't see this in healthy controls, but you see a lot of it in patients with severe COVID-19. And you don't really see very much in patients with mild COVID, which I'll show you in a minute. These antibodies are also Some of them are inducible. So anywhere where you see red boxes, that means that there's an antibody that was not present at an early time point, and a week later, they have very high levels. So interferon epsilon, IL-17F, IL-22. So the way that interferon signaling works is interferon binds to its receptor, activates these two kinases, which then lead to the phosphorylation of these transcription factors, STAT1 and STAT2, move into the nucleus where they interact with a third transcription factor called IRF9, and then this turns on interferon signaling pathways that eliminate viruses. So what we've shown, this is work from Sharia Dhingra in our lab, that if you do flow cytometry and you stimulate cells, peripheral blood cells from a healthy individual, and then you stimulate with interferon and measure 30 minutes later, looking at phospho-STAT1, the cells go from this to this. So they can clearly signal very well. If you throw in healthy control antibodies, nothing happens. There's still very good signaling. If you throw in samples from patients with COVID-19 with this blocking activity, they go back to looking as if they can't signal at all. So very clearly, these antibodies are blocking antibodies. And finally, one of the final data slides that I'll share. This is an example of somebody, this is a patient who had anti-IL-22 antibodies, one of the Marburg patients, no autoantibodies against IL-22 at day 20. At day 29, when this patient became sick again, all of a sudden, very high levels of anti-IL-22. Anti-IL-22 is involved in mucosal immunity. I won't go into more about it now. But again, when you're caring for patients on the wards and you see patients who get sick later on, it could be superinfection, it could be lots of other things, but you should also think that, wow, this could be development of these anti-cytokine antibodies that might be driving these clinical changes. Okay, so the conclusions here, anti-cytokine antibodies are even more prevalent in COVID-19 patients than traditional autoantibodies. We see a lot against interferons and these interleukins in particular. Some are blocking, some are clearly inducible, and we found these in many other disorders. So I think you're going to see that these antibodies are going to be important in many different areas of infectious disease and autoimmunity. So what are the mechanisms? So I don't have time. I want to make sure I leave time for questions. For sure, there are some patients with pre-existing autoreactive B and T cells and their cytokine storm. We think that there's probably molecular mimicry in which there's proteins in the virus that look like our self proteins, and there's cross-reactivity. And then there's some other mechanisms that we're also exploring. And this is one example from Pross's study in which one of the patients who had mild disease at day 0, who was in the placebo arm of their Lambda study, 28 days later, this patient got very sick and was critically ill, developed new antibodies against PM-SCL75, another one of these myositis scleroderma antigens. And we speculate, although we don't know, that there may be cross-reactivity with nucleocapsid or some other viral protein. We're exploring that now. So this is the last slide related to the data. So I like to think now about what are the things we don't know. We probably know much less. There's much more that we don't know than what we do. So we don't know the prevalence of these. We don't know if these are found in other severe infections. I can tell you that they are found in other severe infections. We're putting that data together now. So other non-COVID infections. We don't know if the patients who have these autoantibodies are going to go on to develop autoimmune diseases. Just because you have the antibody doesn't mean you develop autoimmunity. And this is going to require very large epidemiology studies. We don't know if there's any link yet to vaccine adverse events. We're exploring that. We think that there probably are some links. We don't know what happens with patients with autoimmune disease in COVID-19. This is a very open question. So are these patients more likely to get severe disease or not? And is there a link to post-acute sequelae? So I'm going to end now and take questions. I'll point out that Monday is our faculty meeting. So Bob will appreciate that the focus is diversity, equity, and inclusion. I never miss an opportunity to promote physician scientist training, and especially the women who contributed to this study in really meaningful ways. So I'll talk about women physician investigators. I'm going to, for those of you who are interested in physician scientists and the problem for physician scientists, we published a position paper, myself, Brian Kaboka, and several other faculty members in the New England Journal 2 years ago that describes the problem in more detail. There's a really good paper from Catherine Blish that also describes this and talks about the issues with women and with underrepresented minorities. There was a Cell Reports Medicine paper that is particularly well written from a group at Michigan because COVID-19 has really gutted physician scientists' careers over the last year. I'm afraid we're going to lose a whole generation of physician scientists. And in particular, for those who are worried about our women, not just physician scientists, but healthcare workers, there was this academic medicine paper entitled "Who is Caring for Healthcare Workers' Families Amid COVID-19?" And this has been a major problem in the School of Medicine. Who's going to care for the kids when there are so many women serving as frontline workers and as physician scientists? Who's caring for their kids, and what's going to happen to their careers, which are taking a big hit? So I'll end on a positive note. We have this nice high school program that Errol described. This is one of the students 20 years ago. So we now are harvesting all of these students that we started enrolling 20 years ago. About 60% of the high school students, almost 1,000 of them now over the last 21 years, about 60% are women. And this happens to be Marie Hollenhorst. This is me when I was really young and didn't have gray hair. This is Marie now, who's an instructor here at Stanford. Our MSTP directorate has also completely turned over. And when I was the director, I was the last of 52 years of consecutive men as directors. Now Katrine heads our MSTP, and four of the six directors are in Department of Medicine. 4 of the 6 are women. That led to an unprecedented first time ever selection of 80% of the MSTP students this past year who are women. 8 women and 2 men. And then for our MD only physician scientists, we have a PSTP that has 15 women out of 28. So again, a majority. This is the last slide, and then I'll take questions. I think the people on this slide will recognize just how hard the month of March has been for us. Bob selected Hannah Valentine to lead the department effort to put in a PASC grant. We actually put in 2 grants for over $100 million. And UPI led one of the grants, and Melissa and Manisha led the other grant. The other co-PIs of UPI's grant are shown down here. And what you'll notice is so there's great racial and ethnic diversity in the slide, but there's near complete absence of gender diversity. And I think that that's a very good thing and just shows that we need to promote more of the careers of women in science and medicine and to place them in positions of leadership. So let me stop there and take questions. And I apologize, I went over a little bit, but there should be time for questions.
Dr. Ritz, thank you so much. And it just reflects how much you have and colleagues you're working with have going on in this field that's so important to us all. So thanks for that great presentation. As many of you probably see, we have a lot of questions. We certainly probably won't be able to get to all of them. For any of the questions that we don't get to, Dr. Ritz has always been super responsive. Perhaps we'll send you those questions to address and follow up an email. But I want to get into them now. Please also, if you have any questions before you write it, see if there's one similar that you can upvote to help me triage the best ones to answer. I'll start with this first one. Any information or data on available COVID vaccines, effects on autoantibody levels, or even their development? There's been a few questions regarding that. Dr. Morton Boers also asks, any concern the vaccine could trigger antibodies? So really, that's a big question we've been seeing in the theme of questions here, the vaccines and the connection with autoantibodies.
Yeah, it's a great question. So we have data there. I did not show the data today. We have screened 31 subjects from Kari Nadeau and Bali Pulendran's vaccine study all the way out to day 42. 5 of those subjects had pre-existing autoantibodies. The levels did not change at all over the 42-day period following the Pfizer vaccine. So we feel very confident that if you have pre-existing autoantibodies in those patients, none of them changed at all. So we are, Lisa Zaba, Yashar Chachin, Kari Nadeau are doing a study now in lupus and psoriasis, enrolling a vaccine study that's already enrolling, in which we're testing whether these patients, if they have pre-existing antibodies, if they change or if they flare. The data so far would suggest the vaccines appear to be pretty safe, but we would love to test this more formally.
I have Dr. Kutidif also mentioning the same question, but she had 3 patients with autoimmune thyroid disease after vaccination. It sounds like the data does suggest connection, but still something being studied.
That's correct, Joe.
I'll move on to the next question. Dr. Gottfried asked, Dr. Roots, how do positive rates for antibodies compare between patients who COVID positive patients and patients who are clinically asymptomatic? Are there reports of baseline frequencies of these people with antibodies with no autoimmune disorders?
So it's a great question. So we are looking at, so in the convalescent sera from Penn, what we've shown is none of those patients had anti-interferon antibodies. They had far fewer anti-cytokine antibodies and virtually no autoantibodies, other than anti-thyroid antibodies, which are pretty common. And the same thing in process study. It appears that autoantibodies and anti-cytokine antibodies are much lower in patients who have mild disease. We don't yet know about those who have post-acute sequelae.
Dr. Gao asks, are any of these autoantibodies causing thrombocytopenia? Had a COVID positive patient with very low platelets recently.
That's a really good question. Our assay has not looked at antiplatelet antibodies. That would probably require a cell-based assay, but it's a great question and it needs to be looked at.
Dr. Roots, if it's okay with you still, we're going to go about 5 minutes over and see how many more questions we can get to. Dr. Rogers is on our panelist side here, and she had an interesting question. I wanted to hear her talking about the viral, is it unique to COVID versus the viral response? Dr. Rogers, thanks for asking your question.
Yeah, thank you so much, PJ. Great talk. I think one of the big questions in the critical care field is how much are we just studying COVID more, so learning cool immunology, and how much of this is true in sepsis more broadly? Do you think this is a viral thing? Is this specific to COVID and that's why it's worse? Or do you think we'll see that this is broader? I know it's not a question that's been asked, but just your hypotheses would be great.
Yeah, so we have preliminary data that would suggest this is not limited to COVID. So, and now the question is, how broad really is this? Is this seen in, is it just going to be flu or RSV or other things? And so we're working with you now to look at a large number of ARDS samples. My prediction is that we're going to find anti-cytokine antibodies in a significant subset of those patients, and I'm going to predict that some of them are going to be blocking antibodies. So I think that this is going to be, I think that now we have to completely rethink when a sick patient comes in with pneumonia about whether they have circulating factors, genetics or circulating factors that might be contributing to that, because we may need to be immunosuppressing a subset of these patients.
Thank you.
Dr. Roots, thank you for that. Dr. Berry asks a great question. She says, PJ, great talk. We know that women have exuberant autoimmunity compared to men, yet men are dying at a higher frequency in COVID-19. How can you explain this in the context of autoantibodies?
Yeah, so that's a really good question. If you just were to assume that it's related just to the anti-interferon antibodies, there it's 12% of men and only 2 to 3% of women. So really ultimately, I think the question is it women are more likely to get autoimmunity in general. So that finding in and of itself is not what you would expect. You would have expected more women. So I don't really have a good answer for why more men. I don't think it's all going to be explained by antibodies against interferon alpha, though, or secreted factors.
And Dr. Hartman asks, COVID seems to clinically take a turn for the worse after 7 to 14 days on onset, something we all have been seeing. I wonder if the development of autoantibodies explain this delay in clinical severity. So that fundamental question, what's driving the severity in acute illness?
Yeah, I think that could be part of it. I think that there's so much going on pathophysiologically that that's probably not going to be the only answer. I've seen some really good talks by Mark Krasnow, for example, that might explain just how the lung regenerates and the AT2 cells that are the target cells that are infected. So I think it's a really complicated problem, but it would not surprise me if some of these autoantibodies, the blocking antibodies in particular, play a role, like anti-IL-22 might be one, things that are inducible.
Dr. Grimes asked, did you test for antibodies against ADAM-TS13 as an increased risk for thrombosis?
We did not. It's a good idea.
Dr. Rumpf asks, is there any evidence that these autoantibodies developed with COVID-19 infection cause cardiomyopathy?
Yeah, that's a really good question too. On our array, we have a series of antigens from patients that are thought to be targets in myocarditis. So alpha troponin, tropomyosin, troponin. We definitely see MYH6, which I think is alpha troponin. We definitely see antibodies in a subset of patients. But we don't have a good enough if we had a good cohort of myocarditis patients that we could compare, we actually could test that. But we definitely see some reactivity, but we don't have enough clinical data to go back and see if they actually had carditis.
Dr. Vasquez asks, is the autoantibody interferon activity different in obese patients?
So the number of subjects that we've identified who have this is so low that we can't really say. We've got in our study, out of the 147 subjects, we screened about 100 of them, and we had roughly 3 or 4 positive total. So it's just not a large enough number to know.
And next question here, we'll keep going for another minute or so. Do you see these antibodies being used for predicting severity prognosis routinely in the future?
Yeah, that's a really good question. I think that they could be. And the real question is this something that could be developed and run rapidly enough? The sorts of, like, literally we get samples now, we can run, we got samples recently where we got the samples, designed the experiment, ran it, got data within 48 hours. So I think you could do that. And some of these are commercially available tests, some of the anti-interferon tests, for example. They're not difficult to do.
Dr. Lin, long hauler symptoms associated with antibodies. I will say we'll talk more about that next week. Any thoughts, Dr. Utz?
Yeah, that was the focus of my portion and Bill Robinson's portion of the PASS grant that we just put in. So we're pretty actively hoping to study that and to tap into some of this $1.15 billion that the government is throwing at this problem.
Dr. Utz, it's obviously, this is a huge topic, very important. We have more questions come up as fast as we answer them. I'm going to take the remaining questions and send them to you via email, and then ask to respond to the ones that you can, if you don't mind. Thanks for being so responsive this whole past year with anything that's come up. So we really appreciate it. Thank you so much for this great presentation. We have comments from our faculty. Dr. Harm has talked and mentioned, thank you for all the leadership of the MST program, especially with the gender diversity. So thanks for everything. And everybody, thank you for sticking with us this whole time as well. I hope everybody has a great week, and we'll see you next time.
Thanks very much.
And next week we have Dr. Subramanian and Dr. Williams talking more, part 2, clinical aspects of long COVID. Thanks, everybody.